I'd like to welcome you to today's presentation brought to you by the Society of Decision Professionals and the SDP Probability of Success Interest Group, POSIC, which is entitled A Decision Analytic Framework for Bayesian Updating of Probability of Success in Clinical Trials. This POSIC talk wouldn't happen without the efforts of our co-chairs and moderators, Sean Comfort and Eric Johnson. Sean is a Principal Scientific Enablement Director at Roche, leading innovation work supporting pharmacovigilance. He is a neurologist with 20-plus years combined biopharma industry regulatory experience. Dr. Comfort's current work focuses on combining machine learning models and decision analysis techniques to evaluate drug adverse event causality and predict project-based probability of success. Eric Johnson supports drug development decision-making at GSK, where he is helping the new DA practice to grow. Previously, he had done DA work at BMS and for consultancies. He is the co-author of the Handbook of Decision Analysis and a fellow of SDP. Welcome to today's webinar, and Sean, you have the floor.
Thank you, Hilda. So good morning, everyone. So on behalf of the Society of Decision Professionals, both Eric and I are happy to welcome you to our first 2024 POSIG presentation. And we're excited. This is part of a two-part series of presentations. Today with Dr. Tom Keelin, and then on March 20th, we'll have Dr. Sam Savage. And these will explore probability of success estimation, as well as later with Dr. Savage, exploring some of the economic and financial ramifications of these estimations. And as Hilda has said before, today we have Tom presenting a decision analytic framework for Bayesian updating of POS in clinical trials. I know that many of the people on the call have known Tom and worked with him. I see a couple of familiar names there, but for those that don't know him, Tom serves on the Society of Decision Professionals board. He's been a leading DA analyst and practitioner for nearly 50 years, and he invented the Metalog probability distributions in 2016, which you're going to see being used today. And he introduces us to the Metalog Bayesian inference and application for probability of success in clinical trials based on new information. I also think that you'll see that what's done today has a pharma flair. Eric and I and all of us are very much in pharma, but this is a very general technique that can be easily applied whether you're in energy, aerospace, medical devices. So with that, I'd like to turn it over to Tom and have him start us off. Tom, it's all yours.
Well, thank you so much, Eric and Hilda and Sean. I first want to just express a note of gratitude. I looked at the sign-up list for today's webinar. There are so many wonderful longtime friends and colleagues on this webinar. Thank you for joining. I wish we could all get together and have a party. Today's seminar is a decision analytic framework for probability of success in clinical trials. That's different from a statistical framework, and you'll see exactly what I mean. A little bit of background. In 2021, as most of you know on this call, Ron Howard and I published a preprint paper on Bayesian updating using Metalog distributions. It's had more than 2,000 downloads and views together, but that who knows what that means, because we have received very little feedback on this, so we don't really know whether people are actually reading it or not. But nevertheless, Eric and Sean knew this paper was out there and inquired of me as to whether this method could be useful in calculating probability of success for clinical trials, and also the Bayesian updating of probability of success when new data becomes available. These are great questions. We got engaged in a wonderful conversation over the last couple of months, and this seminar is how that has been brought to conclusion. If you haven't seen the paper, by the way, you can Google Keelin and Howard OSF, or I think Hilda is putting a link to it in the chat. So the example we came up with, and this was Eric's suggestion, I think it's a really terrific, clean, simple example where we can actually look at derivation of probability of success. This is going to be a new weight loss drug in phase 1 clinical trials. 16 patients lost an average of 3.1% of their body weight. after 6 months with minimal side effects. Phase 2 trials are underway. A possible phase 3 trial is being planned. That'll have 100 treated patients plus 100 control patients. Average weight loss throughout this presentation is defined as treated patient average weight loss minus that of the control group. So it's placebo effect adjusted. And we're going to use a definition of success, which is that the average weight loss of the treated group is greater. The average weight loss is greater than 3%. You can see here for the 16 patients, there's all this phase 1 data as to what was their percentage of body weight loss over a 6-month period. Typical of a phase 1, there was no control group. The average of this data is about 3.1%. And so for now, we're going to avoid hypothesis tests and other statistical success criteria, but I will say a little later how those can be incorporated within the framework of this analysis. So questions posed by Sean and Eric. Sean and Eric, by the way, generated the data. I had nothing to do with that. I've just been asked to come in as quote unquote a consultant to say how might one calculate or use these methods to calculate probability of success. And there are 2 versions of it I'm going to look at. First, probability of success based on phase 3, based on phase 1 information only. In other words, if you only had the phase 1 information, what probability of success might you be able to derive from that using reasonable approaches? And then secondly, once the phase 2 information becomes available, how can we conveniently update this probability of success based on phase 2 data, that is the phase 2 SOI state of information? And then the last question I'm going to address is, okay, I'm going to give a simple closed form procedure for doing this updating, and the question really is this Bayesian, or under what conditions is this procedure that I've presented Bayesian? And we will address that at the end before the Q&A period. So let's get into this. A natural starting point would be to consider the distribution over weight loss for the next patient or next patients until new information becomes available based on the phase 1 data. So for now, if we assume a control of 0 and just take these 16 data points, put them on a graph, fit a metalog to it using ordinary least squares as just a 3-term metalog, then we end up with actually, it looks like quite a good representation of that data with the metalog. And if you look at the right, the associated density function is a bit right skewed. We have the success criterion of 3%. The mean of this distribution is 3.1%. Well, what would we make of that? Based only on this information, how would we calculate or even think about calculating a probability of success, assuming the next trial, suppose phase 2 is skipped and we just go directly to phase 3, it would do a 100 patient treated patient trial with a control group. How would you think about that probability of success? Interesting, I've been around this space for a long time, but I've not done this kind of calculation before the question that Eric and Sean asked. So there's a big if here. If this probability distribution that I just showed, that is this metalog fitting through the data, if that were representative of the entire population, then the phase 3 probability of success could be conveniently calculated by simulation. Well, how would we do that? Well, we've got the probability distribution. It's easy to sample. Metalogs are easy to sample from, even in closed form. And so what you do is you simulate 100 patient trials. You draw 100 patients from the 100 samples from the distribution. You find the average weight loss for those samples. If it's greater than 3%, then that's a success. If it's less than 3%, that's a failure. And if you simply did that, the observation from doing that is that in 68% of the simulated 100 patient trials, the average weight loss is greater than 3%. So therefore, we would say the probability, phase 3 probability of success based on phase 1 information is 68%. Now, that's an appealing way to do it, but it turns out that this is not a correct answer. And it's interesting to consider why it's not a correct answer. The reason it's not a correct answer is it becomes illustrated if you simply do a thought experiment. The thought experiment is, what if we increase the number of patients in the trial from 100 to 1,000? You do exactly the same calculations, exactly the same simulation, except each patient trial has 1,000 rather than 100. Well, the probability of success in that case would drop from 60, or would increase, I'm sorry, from 68% to 92%. Well, why would that happen? Why would that happen? Well, because this original distribution we have based on phase 1 data has a mean, and by the central limit theorem, the higher number of patients that you have in your trial, the closer the the simulated results will be to exactly that mean. The mean is just slightly above the success criterion. So it means that if you go to more and more patients, the more patients you have, the higher is the probability of success in the clinical trial. Well, in the phase 3 clinical trial. Well, that clearly doesn't make any sense, because you're still only based on phase 1 information. You couldn't, if you did a 1,000 patient trial or a 10,000 patient trial, you'd be approaching 100% probability of success in phase 3. It makes no sense at all, because you really only have this phase 1 information. So, and you could do the same thing even if you assume, for example, a 0.5% placebo effect just as a plug-in. Well, then that would shift everything down. Your probability of success in the 100-patient trial would be only 3%, but then in the 1,000-patient trial, it would be virtually zero. Well, again, it makes no sense to think that a phase 3 trial based on phase 1 information, which was relatively positive information, could have a zero probability of success if you increase the trial size. Well, the fatal flaw in this kind of analysis or way of calculating probability of success for the phase 3 is we have assumed this distribution is representative of the entire population. And all these things I just stated, if this distribution that I showed at the beginning, back up here, if this were in fact representative of the entire population, then these results I've shown would be correct. And the more trials you have, the more your probability of success would go to either 0 or 1. However, there could be a lot of different distributions over the entire population weight loss. We don't know what that distribution is. We'll never know. The idea is that we should be able to learn about that by the data that we have observed. But for example, this distribution right here is the one I just showed, but that might not be the actual distribution for the entire population. The actual distribution for the entire population could be this one, which is more symmetrical, or it could be this one that has a long left tail and a very short right tail, or it could be this one that has a long right tail and a short left tail. We don't know particularly based on just phase 1 data, which of these distributions might be applicable. And every one of these distributions has a mean. And some of those means may very well be greater than the success criterion, some may be less. And so the real question is, how can we appropriately generate a probability distribution over the possible probability distributions for the weight loss, for the weight loss distribution? There are a couple of different ways of doing that. The way I'm going to do it is consistent here with what was in the 2021 paper. The idea here is we have our probability distribution over weight loss for the next patients based on phase 1 data. That probability distribution is parameterized just by least squares. It's parameterized by a 10, a 50, and a 90 point. Now we could use 5, 50, and 95. It wouldn't make any difference, but let's just use 10, 50, 90 for now. And these 3 points here control the location and shape of the distribution. And as of the time of phase 1 data, where we have very little information, we actually don't know. I mean, it's reasonable to assume, I think, that these 3 points would be the center of a range of uncertainty about the way they're ultimately going to come out. But if you gather more and more data such that you eventually had the entire population, the distribution for the entire population, you might find that that median, while it's 2.4% in the base case, that median might actually range from 1% or less than that up to, you can look at this distribution here, up to 4 or 5%. You don't know which median you're going to get for the entire population when you sample more and more data. Same thing for the 10% quantile. You don't know which, and that's even a wider distribution because there's more uncertainty inherently over the 10% quantile than there is over the median. That's why that distribution is a little bit tighter. Same thing for the 90% quantile. It could have a range on it. And depending on which samples you get, if you were to sample from this and furthermore, there's some correlation between these. In other words, these are not three independent, even though these distributions are symmetrical, they're not three independent probability distributions. If the whole population, it turns out, ends up over here with a higher median, then presumably there's a positive correlation between that and where your 90% quantile is. And it may be also with where your 10% quantile is. So there's a covariance matrix among these. I've just shown the marginals right here, but a key way, and this really is the basis for Bayesian inference, is making the parameters of a model uncertain and then learning about those parameters from the data you observe. So for each of these, if I drew this as the median from here, and if I drew that as the 90%, or let's say drew this as the 90% quantile, I might very well get a distribution over the entire population that looks like this. Other draws would lead different things, leading to a range of such distributions for the population. So that now gives us a method, a procedure for calculating probability of success for a given state of information. So here we are. What is this? What would this success? What would this be? This method? Well, generate a sample of the weight loss distribution from the distribution over the parameters of the entire population distribution. So I'm going to take a sample from this multivariate distribution over the three quantiles. From that, I'm going to get a metalog. From that metalog, I'm going to, conditional on that distribution, generate an n patient sample, let's say 100 patient sample of weight loss. I'm going to apply my success criteria to that sample. That is, the average weight loss is greater than the 3% success criterion is success. I do that n times, like 1,000 or 2,000 times. Probability of success is number of successes divided by the number of trials. If we did that based on the unadjusted phase 1, we end up with a probability of success of 52%. And you can see here, here's the histogram of the average weight loss for each of the 100 patient samples. And in the middle of that, you can see as a success criteria, on average, the probability of success just a little bit greater than 52%. If you did the other case, assuming the 5% placebo effect for phase 1, you end up with probability of success around 28%. So this is a way to do it. Now, I would like to, and I think this is a relatively robust procedure for calculating probability of success of a phase 3 trial for a given state of information, in this case, phase 1. I would note here that this success criteria, it doesn't have to simply be an average is greater than a target. It could be any function whatsoever of the patient data. So you could put in here statistical significance criteria, or even a more complicated criterion, such as more than 50% of the patients lost more than 4% of body weight in excess of placebo. Well, once you have the sample of patient data, all of these things could be easily calculated. And so you could determine success or failure from that and still use exactly this method. So that gives us a method for calculating probability of success given a state of information. I've often seen that people will try to shortcut this, and some shortcuts work, some shortcuts don't. I've heard some people say, oh, well, if you really have all these possible distributions over the ultimate, the whole population weight loss, why don't you just average those? And there's, when you use linear opinion pooling in In this case, I've used the Vincent average, which is basically averaging the cumulative distributions instead of averaging the PDFs. There are lots of arguments about what's the best way. The latest information, based on a recent Management Science article, is that this Vincent averaging may be better. That's also discussed in my 2021 paper with Ron. But the key point for this presentation is that the choice of an averaging method is arbitrary, no matter what you use, linear opinion pooling or the Vincentization, which is this yellow one. No matter what average you use, that average is going to have a mean. And therefore, what that means is the probability of success will go to either 0 or 1 as patient sample sizes increase, which, as I've mentioned, it should not. So this is not a good way to make a shortcut to the above procedure. In contrast, if you're going to use the mean as being greater than or equal to a success criterion, then you could just, instead of averaging this distributions, you could calculate the mean of the distribution after you sample from the entire patient population. And that would actually work pretty well for this success criterion. Here's an overlay of the means of all of these distributions put on top of the results of the 100-patient trial average. And it's basically, it's 27% if you use the mean, and this is for the adjusted phase 1 data where we've used a 0.5% placebo effect. And it's 28%, which is a very, very similar number, obviously, if we did the entire sampling of all the patients. So basically, if you're using this kind of success criteria, the larger number of patients in phase 3, the more reliable is this proxy. However, the basic way to do it is to just do this, as I've discussed. So now let's turn to, so I've now shown that the probability of success, I've shown how to calculate a probability of success for phase 3 based on the phase 1 state of information. The question is, how can we conveniently update this probability distribution based on phase 2 data now? And Eric and Sean, and particularly Sean, went to work on his computer, generated some phase 2 data. I had nothing to do with this. He just said, here it is. And that's the way clinical trials go. You get your data back, and whatever it is what it is. And that's what you need to deal with. Anyway, Sean generated 2 cases for illustration. One is what I'll call a favorable case. And in the favorable case, you can see all this data here. There were 19 patients, I believe, and then plus another 19 in the control group. The control group average was 0.25%. That's the placebo effect. But the overall average, if you average the 19 patients and subtract off the control, is 3.7%, which is clearly a favorable case. That's certainly better than even your phase 1 data. He also generated a phase 2 case where the overall average is a little bit less than the success criterion. And I'll use these 2 cases to, one, show how one could update one's state of information, and then 2, how you would update the probability of success based on the updated state of information. Now I'm going to start with, I'm going to start by going to the end, because I think that's the easiest way to understand it. What happens if we have 19 new data points in addition to the 16 we had from phase 1? Well, now we've got 19 and 16. We've got, whatever that is, 35 data points. So with the more data you have, the tighter will become these probability distributions over the metalog parameters. That's not surprising. If you had 1,000 data, then that would be enough to make these distributions very narrow over the parameters, right? You'd be narrowing in far more on what the median is, or what the 10% or what the 90% is. So basically, if you have this prior distribution, which is the same one I showed before, if you're just based only on the phase 1 data, and then you get all this new information for the 19 patients in the control group, then you're going to end up with tighter distributions, number 1. But number 2 is the center of those, the quantile parameters of your updated metalog may very well change, where in this case, the mean of the distribution over the median is actually quite a bit higher. And the 90% quantile is higher, and the 10% quantile is just a little bit higher. Well, that's just based on the nature of the data. But if you had this data and these updated distributions, and then you sampled from these, and they're still correlated distributions over the parameters, and then you did our probability of success calculation for each sample, let's say you did that 1,000 times, you would end up with this probability distribution over, this is the histogram of the average from the 100 patient clinical trials. Most of those trials are greater than the success criterion. The probability of success in the favorable case is about 80%. So the two key questions here, given that we're going to get to this as an end, two key questions are, well, where do I get this updated mean from? And where do I get this narrower covariance matrix from? How could those be calculated? Well, let's start with the updated mean. There's what I would call an appealing method, because it's so simple, would be to combine representative prior data, and I'll say what representative means in a minute, with the new data. So combine the prior data and the new data and define the updated mean as the metalog parameters fit with least squares to the combined data. How simple is that? In other words, here are the original data points from phase 1. Here's the new data. These light blue dots, that implies a new metalog. The new metalog has these 10, 50, 90 quantile parameters, and those become, naturally, it would make sense to have those be the mean of the 10, 50, and 90 quantile parameters of the posterior distribution on the distribution of the parameters. So that's actually pretty simple, and that's the method I'm proposing. Now, I said I would say, what does it mean? What representative prior data would we use? Well, there are a couple options for that. One option is to use all the prior data. Here's all the prior data. You could stick all those in, and that would be just fine. There's nothing wrong with that at all, and it would make sense. However, what happens in so many clinical trials and clinical trial situation is that something changes, and you'd like to have another option. Something changes because, well, for example, the phase 2 trials have a control group. The phase 1 trial did not have a control group. Maybe the definition of the patient population has changed in some way, or for any number of reasons you might believe maybe the phase 1 trials were done in a different country with different population characteristics. There are any number of reasons why you might believe that the phase 1 data is not as relevant to the final result as the phase 2 data. And so the option for that is to assess an equivalent sample size. We'll call that N0, and we're going to use N0 data with the same shape and location as your prior data. So for example, if you believed that it would take 2 phase 1 data to have the same weight or the same information content as 1 phase 2 data, you might use an N0 of 8, which is what we've used in our base case. And in that case, we just come up with a new synthetic prior set of data, which are these 8 data points, and we just take them off of the prior metalog distribution over entire population weight loss. So that's a simple way of doing it, and you can choose either one or the other in this method that I'm presenting. Now, what about a covariance matrix, and how might we update that? Well, specifying and updating the covariance matrix is even more convenient, according to a set of equations that's in the 2021 paper. But first of all, there's nothing in this method that requires assessing a covariance matrix, which is actually a fairly difficult task to do with any reliability. In this method, the covariance matrix depends only on 2 things: an initial width called sigma and the total number of data, n, okay? And the total number of data is the total number of combined data and the total combined data, including the prior data and the new data. So let's just talk about how it depends on this total number n first. Well, the total number n basically specifies the width of your distribution over parameters. If you only have 20 data, then naturally this distribution over parameters is very, very wide. If you had 200 data, it gets narrower. If you had 2,000 data, by then you've got a very good fix on what is likely to be the median within a very narrow range of the entire population, as well as the 10 and 90% quantiles. In fact, with this method, the variances and covariance, in fact, the whole covariance matrix shrinks in proportion to 1 over n, which is not only intuitive that it would go down, but it actually goes down at the same rate as does the variance over the average in the central limit theorem. So that's convenient. And then the other parameter is the initial width sigma, and you can think there are many different ways of interpreting sigma, but one of them is that it's just simply the width of this initial distribution, prior distribution over the median. If you made that whole thing wider, then you'd have something that looks more like this, and you'd have a much wider range of variability in the parameters you're sampling from. And if you made it narrower, you'd be expressing that you have you're more assured of the parameters that constitute the prior. So there's a very simple method. It's all closed form, few matrix manipulations and so forth for updating the covariance matrix. And what I've said now up to this point is, okay, suppose we have prior information. We've just used these very simple intuitive methods for updating both the mean and the variance and the covariance of the distribution over parameters. And what I said previously is that when we sample from those, we found that we had basically an 80% probability success in our phase 3 clinical trial based on the updated phase 2 information for the favorable case. Now, what I'd like to do is explore a little bit in my last few minutes here what is the sensitivity of I've got various parameters in these models. A wonderful way to debug a model is to see whether, like that phase 3 probability success, how much does that change based on changes in any of the underlying assumptions. So when we go from the favorable phase 2 data to the unfavorable, well, that's a big drop. That unfavorable data is pretty unfavorable, and it brings you down to like a 28% probability success. What about the success criterion? My goodness, if you change that from 3% to even 2.8, which is a pretty small change, your probability success goes up to nearly 90%. And if you change it to 3.2, it drops down to like 65. What about the strength of prior N0? Here we used 8, but if we 0 would mean we completely ignored the phase 1 data. And if we did that, the probability of success goes up to nearly 90%, which is not surprising given that the phase 2 data is more favorable than the phase 1. But if you used, on the other hand, all of the phase 1 data, then your probability of success drops to like 68 or some such thing. The phase 2 placebo effect, if that were to change by 0.2%, increase by 0.2%, your probability of success drops to like 70 and so forth. You can work all the way down here. The initial width sigma, the wider the initial width, you move it from 2 in the base case to 3, you're just going to have less. You're going to have less assurance of where your parameters are, and because of that, you've got a wider distribution over that, and because of that, you're going to have a wider range of results on the mean and a less likelihood of coming up with a successful clinical trial. And as you increase that even further, it would go down here to almost 50% or something like that. Number of patients in your clinical trial, we used 100. If you moved up to 500, that would increase your probability of success a little bit. If you went down to 20, because your trials would not be as definitive, it would go down a little bit. There's a phase 1 placebo effect. and so forth. Number of simulation trials makes almost no difference. I used 1,000 in the base case, up to 2,000 wouldn't make any difference. Random number seed makes no difference. So anyway, all of these sensitivities, which are really all of the parameters in the model, are looking pretty sensible as far as I can see here. So the last question Sean and Eric posed is, okay, well, that is a disarmingly convenient procedure. I mean, we've done it all in closed form. It can be done in an Excel spreadsheet, as Sam Savage is going to show in our next POSIG seminar on March 20th. The key question, though, is, well, isn't that just, I mean, it's a sensible procedure? Yes, but is it Bayesian? And under what conditions is this procedure Bayesian? Well, it turns out that there are 4 conditions, and these are outlined in the 2021 paper. But the first condition, and if these 4 conditions are all satisfied, then the above updating procedure, one can show that that is in fact Bayesian. And what are those conditions? The first is the target variable distribution. In this case, the distribution over weight loss is a quantile parameterized distribution, which implies that the quantile function is linear in its parameters according to this paper down here in the lower right, published in 2011. Well, if you have a quantile function that's linear in its parameters, then we can make a few further assumptions and get all these simple closed forms. The second assumption is the distribution over the parameters, that's this distribution that I've been showing, is multivariate normal or student t. That doesn't seem to me to be such a bad, I mean, the distribution, the overall distribution here can be skewed, but the distribution over parameters, I don't really have much reason to believe that it should be skewed one way or the other for a given parameter like the median. Probably it's okay. You can judge for yourself, but an assumption that helps make this Bayesian is that distribution is a multivariate normal or student t. Third, sample errors, quote unquote, are normally distributed with standard deviation sigma and mean 0. This is the standard assumption. It's even called the standard error model in statistics that is underlying multiple linear regression. It implies that the likelihood function is a product of normal distributions, and that combined with the distribution of parameters being multivariate normal or multivariate student t means that the distribution over the parameters is a conjugate prior. And if sigma is certain, it's a multivariate normal. If sigma is uncertain, it's multivariate student t. You can work through all the math as in the 2011 paper. But the big deal is you've got a conjugate prior now, which means you can do the updating in closed form, except you can't quite do it in closed form without one more assumption that Ron Howard and I put into the 2021 paper, and that is that the prior data contains all relevant prior information. And so this, whatever you use for prior data, there is no prior to that prior. There is no additional information contained in the prior beyond what's expressed in this data. And that enables these what in many ways are complex equations that are implied for the Bayesian updating. It enables those to simplify, and a lot of terms drop out, and it enables those to simplify to the point where you just use metalog regression to update the mean, and you can narrow the covariance matrix, as I mentioned. Now, one last caveat here. George Box came up with this: all models are wrong, but some are useful. There is a problem for a small fraction of samples from this multivariate normal distribution, or multivariate t, and that is a small fraction of the samples of the distribution over the parameters is infeasible, or not even a probability distribution at all. You could draw a sample from here such that the median is less than 10% quantile. That wouldn't make any sense. So we discard those kinds of samples. But other than that, as far as I know, this whole approach is Bayesian and works. So and basically simple closed forms, implementable Excel, and so forth. So that completes my my overview and introduction here. Thank you all for joining. It's just fun to do this with so many old friends and a lot of new friends and potentially new friends. And at this point, I'd like to turn it back over to Sean, who is moderating the Q&A period.
Thank you, Tom. It was very good. Enjoyed it. We did get a few questions that I took an attempt to answer, but some of them I think are questions others would like to hear about too. And one, I know that you've used metalogs for subject matter expert characterization. So one individual's asked about, you mentioned the distributions to those belong to people, SMEs, who, and I pointed out today, the data we derived it from the synthetic data I generated. But could you just speak a little bit about using SME judgment, that you can also build metalogs from that as well if you don't have data?
Yeah, sure. Yeah, that's a great question. Yeah, if you don't have data, and let me go back a bunch of slides here. There's no reason at all why you couldn't. Here it is. There's no reason at all why you couldn't start this method with simply an assessment of your 10, 50, 90 quantiles from a subject matter expert. If you had no phase 1 data, you could certainly do that, and all the method would proceed exactly as I've described. And in that case, what you'd do is you'd start off with your assessment from a subject matter expert, or maybe a pooling of data from multiple subject matter experts. And again, that pooling of data from multiple subject matter experts is covered in our in the 2021 paper. But if you start with this, all right, and then use, you know, an N0, which basically indicates the strength of that data. And again, that could be assessed from subject matter experts. Do you need three? Do you need one new data point? Is one prior data point? Is 3 prior data points worth 1 or 2 new data points, and so forth? You could assess these 3 quantiles and the N0, and you're off and running, and you use this whole method.
Great, thank you.
Can I ask a follow-on question to that, because I think that's a great question, and it ties what you're doing here, which is related to the assumption that you and Ron stated, which is that the phase 1 data is a sufficient characterization of your prior, that you knew nothing else besides it, which of course is not quite realistic, but maybe it's a good enough working assumption. But let me ask, in your tornado diagram, you showed sensitivity to sigma and mu. Now I'm trying to trace back.
Okay.
Where are those parameters that you assessed from an expert? And if so, can you talk about assessing those?
No. The sigma mu distinction here, that's a great question. Thank you, Eric. The sigma mu distinction is just back to, if you were trying to do this simplification that I mentioned, which would be, instead of doing 100 patient samples, instead of doing that, if you simply generated a sample distribution, you sampled from your distribution of quantile parameters, and then, like in metalogs, you can just use the mean equals A1 plus A3 over 2. The mean is a trivially simple calculation. And if you use that instead of actually doing a simulation over your 100 patient trials, then you would get almost exactly the same answer as what I was referring to. And you wouldn't have to do the simulation over 100 patient trials, because what happens is you're just assuming, if you use the mean, that your phase 3 trial is going to be large enough in terms of number of patients that the mean you get out of it will be pretty close to the actual mean of the distribution over the entire population. So that by the way, this whole method would not be true if you're using statistical criteria, hypothesis testing, other things. So I think of it as more of an asterisk on the whole method rather than a fundamental. It's just there more for interest.
So Tom, we've gotten a couple of questions that really follow along with this and what Eric has asked about. One was really pretty fundamental question. I thought I'd pass this by you. does the Bayesian procedure imply what that N0 prior size should be? Basically, what is the effective sample size? And when would it be unreasonable essentially to just use all of the phase 1 data?
What is it, Sean? I'm sorry, I couldn't quite hear your question.
So does the Bayesian procedure or Bayesian inference imply what that prior sample size should be?
Oh, no. Does the Bayesian inference procedure imply what the N0 should be? No, it does not. And let's talk about that for a second. That's a great question, in fact.
Then Dimitro asked, would it be unreasonable to just set it equal to the entire phase 1 data size, which I think he created at 16 or something?
Yeah, and that's a good question from Dimitro too. Let's see. Let me find my slide on. Okay. All right. The question is would N0, that is, if I just picked these 8 data points, used 8 of these data points here, is that assessment implied anywhere in the Bayesian updating procedure? And the answer is no, it's not. And that's important. It's an additional assessment that you need to make in order to do Bayesian updating. If we just went with this distribution here based on the phase 1 data by itself, this distribution contains 2 of the 3 key pieces you need for Bayesian updating. This distribution contains the shape and the location of the distribution over the, let's say, the patient, the next weight loss for the next patient, shape and location. What it doesn't contain is any information about how strong is that distribution, how strong that distribution is relative to new probability or relative to new data that you'll get later. And that's what that N0 basically is. You have to, as part of Bayesian inference, somehow have to specify how strong is this prior distribution? How many data does it represent, for example, relative to new data you're going to receive in the future? That's why we use the N0. Going to Dimitro's question, would it be reasonable to simply use all of the phase 1 data? Yes, it would be if you believe that phase 1 data is equally valid and equally applicable as any of the new data you get in the phase 2. In other words, the experiments were done the same way, the populations were pretty much the same. You believe that the phase 1 data is equally valid as the phase 2 data, well, then you would use this one. If you believe it's less valid, or you could even use more valid, you could use an N0 of 30. I don't know why you would do that, but anyway, if you think that the strength of the prior data is different from the strength of the new data, then you would use this method.
Great. We're actually getting a couple of questions along this. I'll ask one more and then ask Eric if he has some he wants to bring up. Again, one question that we had that just goes to your metalogs. Someone asked, do you have to use the P10, 50, and 90, or could you use more data?
Could you use the what? I'm sorry.
To that. Someone asked if you had to use just the P10, 50, and 90 symmetric triplets to define your metalog curve, or could you use additional data? You could
use, well, the 2 answers to that question. First, no, you don't have to use 10, 50, 90. You could use 25, 50, 75 if you wanted for a 3-term metalog, okay? You could also, there's nothing about this that assumes 3-term metalogs. If you wanted to have more shape flexibility and more precision in shape, then you could use 4, 5, 6, 10-term metalogs. I've applied this procedure in the 2021 paper to even as many as a 9-term metalog, where these red points, there are 9 control points on the distribution, and it's a multivariate normal over 9 variables, which is where each of these control points exists. So the answer is no. There's plenty of flexibility there to use either additional shape flexibility or use a different definition of where these 3 quantiles reside.
Thanks, Tom. Eric, do you have a question you want to bring up? Any that you've seen?
Yeah, I mean, there was one in the chat, in the question Q&A, that said, essentially, do we need to partition our data into a training subset and a test subset the way they do in machine learning?
No, you don't. All we're doing here, we're not trying to train a model in a predictive sense, which is what happens in machine learning. And we're furthermore not trying to do what is typically done in classical statistics, which is to use a deterministic model with uncertain parameters and then try to update those uncertain parameters based on new data and make predictive forecasts. That's not quite the way we're approaching it here. The way we're approaching it is just from a fundamental probabilistic perspective. What's your prior distribution? What's a reasonable prior distribution of your data? What's your strength of prior? Let's put that together with the new data. Let's fit a new metalog to it. And what's your uncertainty over the range of parameters in that case? And once you have that, you're good to go with the method of probability success calculation that I put forward.
One more question about the distributions. When you create prior representative data, how important is it that it is symmetrically situated around the median? Does it need to be equally spaced? I think that's referring to the triplets, that wondering if you could have them maybe a P40, a P50, and a P90. I'm not sure, but that's the question.
Sure. If that's the question, sure. Yeah, it doesn't have to be. It doesn't have to be symmetric probability trace. It doesn't have to be 10, 50, 90. If you really want to do a P40, a P60, and a P75, or a P95, whatever might be relevant in your situation, that would be totally fine. That all works in closed form.
I'm going to tag team with Eric. Eric, you want to pick another one from our list there?
Well, there was a comment, I think, that said on slide 14, the treated average is about the same, about 4%, but then the placebo effect differs very substantially. And I think Tom's answer is, well, the data is what the data is. I mean, is there more to be said here?
Yeah, I would say one more thing. I think that question points out a simplification that I made in this model, that a more sophisticated model, you may want to do it in more detail. I've simply subtracted the control group average from every patient here, and then I've dealt with treated patients minus the control group average. In a more sophisticated model, you could have a separate probability distribution over what the control group is going to show, right? And that would require different assessments of what that probability distribution is, and you could do your simulations based on that. From the standpoint of all the methods I've shown here, adding that additional modeling detail is kind of a fielder's choice, depending on your situation, whether you feel like that would be helpful. But it really it doesn't impact any of the methods that I've shown here, but it could be a refinement, let's say, in the way we do the calculations. And in fact, control groups, as you guys well know, do come up with surprising results sometimes.
Sometimes very surprising results. Tom, the work you've done here, I know that you've used some different software for it. Can this be implemented in Excel?
Yes, it can. In fact, I have two models of this. One is in Excel, one's in Mathematica. But Sam, basically, we've got another follow-on March 20th with Sam Savage, and he will work in Excel, and he will have this whole system implemented, including sampling from the multivariate normal and whatnot. He'll show how to do all that stuff. So that's anyway, that will be coming next time in the March 20th webinar.
Thanks, Hilda.
Well, thank you. Ladies and gentlemen, this concludes today's webinar. I'd like to thank our presenters and thank you, our attendees, for your contribution to today's event. I also like to remind you that you can find this webinar and all previous webinars in the member-only section of the Society of Decision Professionals website at decisionprofessionals.com. We hope to see you in the part 2 of the POSIX presentation on March 20th with Sam Savage. Watch for the announcement. Thank you.